this LCMS technique. The first separated one followed by the ionization and separation of the ions on the basis of their mass to charge ratio. The separated ions are directed into a photo or electromultiplier tube detector or Faraday detector and identified based on the M/Z ratio. So next slide, sir. The various LCMS includes various extraction procedures during the drugs and metabolites. So extraction procedures includes solid phase extraction, liquid extraction, denaturation. That is for the operation of LCMS technique, there are different extraction procedures depending upon the nature of the components. We are having the different extraction methods for the sample preparation. They includes the regular one we are using is the solid phase extraction, and in some cases liquid extraction based on the nature of the drug substance, and which includes the a little bit rare method of the extraction, which includes depending upon the nature of the sample, includes protein precipitation, that is denaturation. Generally, in the case of LCMS, column or sample preparation includes fast LC column, that is short column we are using, our micro level column of 20 to 150 mm diameter column we are using for the sample preparation. So generally it consists of the analyte concentration and removing the compounds that can cause the background ion or suppress the ionization. So on the column concentration, to increase the analyte, the main sample preparation includes column concentration to increase the analyte concentration. So desalting is there to reduce the sodium and potassium adduct that is commonly occurs in the type of the preparation method called electron spray ionization. So there are different interfaces for the LCMS. That is, it connects to the LC and MS as difficult to interface a liquid chromatography to a mass spectrometer, causes of necessity to remove the solvent. The common interfaces are ion sources, which includes electron spray ionization, thermospray ionization, that is TSI. Next one is atmospheric pressure chemical ionization, APCI. And fourth one, which includes is atmospheric pressure photoionization, chemical ionization, photoionization, electron spray ionization, thermospray ionization. That is electrosource and thermosource, whereas chemical source and photo source as a ionization methods. The commonly used ionization method for the ion source of the sample preparation is electron spray ionization. Next slide, sir. So electron spray ionization is a method for the solution phase of the ions into the gas so that they can sample in the mass spectrometer. So this ionization is the best suited ionization method for the ionic compounds with the high polarity. So that is, more polar compounds can regularly undergo this electron spray ionization. When analyte is transferred from solution to gas phase via this ESI, so solution undergoes three major processes. The common process, either of the one process which occurs in the electron spray ionization includes production of charged droplets, Coulomb explosion, and formation of the gas phase ions from the charged droplets. That is based on the kV volts, that is 3 to 5 kV volts. So sample solutions are tip of the capillary tube which is attached. It will apply to the voltage of the 3 to 5 kilowatts. Mainly the nebulizer gas we are using for the flow from the outside within the same polarity as the applied voltage. That is, it provides the softest ionization method among the ionization methods, which mean that it can be used for the highly polar compounds and the least volatile compounds. Generally, volatile compounds we can move for the GCMS methods, the least volatile compounds and thermally unstable compounds. So we will proceed for the electron spray ionization. So this is a diagram of the occurrence of the electron spray ionization, that is spraying nozzle, analyte molecule, that is it is evaporated based on the charge, and either of the one method, and finally the naked charged analyte will get the droplets as a packets. So based on the positive mode or negative mode of mass detector, we are So seeing the sample, that is the charged droplets are produced by the analyte solution. So highly voltage capillary, that is electrochemical reaction of the solvent occurs, which causes an electron flow. Mainly the condition which undergo the stable liquid can exist with competing forces of an electrofield, the surface tension of the liquid. That is, all the charged droplets are driven away from the each other by the Coulomb repulsion and move along the direction of the electric field. That is based on the direction of the electric field. So the droplets, charged droplets are driven away from the each level. That is, we can see in the diagram, electron spray ionization diagram. That is the spray needle. So based on the Taylor cone, the electron spray ionization droplets are packed as mass spectro compounds, and the solvent and the nebulized ions will be coming to one side. That is thermospray ionization, the another method. So these consists of the two types, real TSP ionization and discharge electrode for the external ionization, and the that is the discharge electrode for the external ionization and the repeller electrode. That is the, this is a type of ionization which can based on the thermostable substances or the sprayed ionized in the LCMS method. So the other method includes atmospheric pressure chemical ionization. This we can call as the APCI method. So the primary application in the area of ionization of the low mass compound. Generally, low mass compounds are applied to this atmospheric pressure chemical ionization method. That is low and moderate polarity compounds. That is highly polar compounds are generally done with the electron spray ionization, whereas moderate polar compounds and which are moderate polar compound which are stable, we can apply it with the thermo ionization, whereas low and moderate polarity compounds can be applied with the atmospheric pressure chemical ionization. That is, so this is not suitable for thermally unstable compounds mainly. So this is for the some of the compounds which are so stable for the thermal, then only the APCA method is applicable. Mainly this the diagram which explain about this one includes APCA is, that is a liquid through nebulizer. We are getting the corona discharged electrode through heater, high vacuum is created, and this will cause the thing as atmospheric pressure chemical ionization. Nebulizer gas through heater, desolvation and drying gas, and we are getting the ion source. And next diagram will explain the transfer of ions to the mass analyzer, that is through capillary evaporation chamber and nitrogen gas, nebulizer gas, and this charged ions are, that is M by Z ratio based ions are released as the source. So when we compare the, there are four methods, we discussed about the four methods of the ionizations, which includes ESI, APCA and all this. The major methods includes electron spray ionization and the APCA. If we compare in the next slide, we compare the differences between the electron spray ionization and the APCA method. That is naturally these are the two important methods. Still, electron spray ionization is the very liquid method of ionization for the source of the LCMS. So it is used generally for the natural substances, biological macromolecules, and pharmaceuticals. So we are using the regular ESI method. So whereas for the low mass compounds, we are applying the APCA method. So we discussed that polar compounds generally undergo electrospray ionization, whereas low and moderate polar compounds gone through the APCA method. Generally for the ionic compounds, we are proceed for the electrospray ionization, whereas for the fat soluble compounds generally will proceed for the APCA. So least volatile compounds are thermally unstable compounds are through electron spray ionization. So this is generally APCA method is not suitable. That is unstable for the thermal compounds. So that is ionization is through protonated molecules or the deprotonated molecules by the ESI method, whereas the regularly this APCA method is generally done for the ionization through ion molecule. So whereas the other method includes APPA, atmospheric pressure photoionization. So APCI is chemical ionization. APPI is atmospheric pressure photoionization. That is chemical ionization as a modification, photoionization. So in this, eluent is vaporized using the heater. The diagram is showing that vaporized using a heater at atmospheric pressure. The resulting gas is made to pass through a beam of protons, discharge lamp, UV lamp, which ionizes the gas molecule. Generally, APPI method is includes the analyte by the irradiation technique. In this irradiation technique, so the short wavelength vacuum ultraviolet, that is short wavelength vacuum ultraviolet light irradiated through this method as a photoionization, and it will analyze this analyte's ionization energy is lower than the photon energy. The analyte ion that was ionized due to the photon may receive a proton from the hydrogen in the solvent and protonated cations. This ionization technique is generally so sensitive techniques. So this includes the sensitive ionization with low or moderate polarity compounds. Just now we discussed that moderate polar compounds are done with even APCI method and also APPA method. So in the diagram, what we previously observed is that we are having the different, as a part of LCMS technique, we are having the different ionization methods as a four methods: thermo methods, electron spray ionization method, chemical ionization method, and photoionization methods. Later on, all these ionization sources are separated with the mass to charge ratio, and this is detected by the mass spectrometer as a detectors. There are three types of detectors which are observed in the CMS, which includes photographic plates, Faraday cup, and electron multiplier tubes. So photographic plates, these are high resolution, and these are generally used in the electronic devices which detect the ions based on the M by Z value. Whereas Faraday cup is the ions which are directed, and the signal produced is very stable and reproducible. That is, the signal which is produced in the detector is stable and reproducible. Generally, spectrometers which are having the quantitative data is most important for this Faraday cup metal cup. In this metal cup, all the ions are directed, and the signal produced is very reproducible signals. Whereas regularly we are using these two detectors, whereas the third one is electron multiplier tubes. So in this, current can be measured, that is, can be measured generally accurately. So on the ion strikes detector can be measured. So ion strokes, so the ion strokes which are having the surface of the electromultipliers or two electrons are ejected. This process continues until the end of the electromultiplier end is reached, and the electric current is analyzed and recorded with the electromultiplier surface. So whereas some of the points of the LCMS are generally, this is done for the molecular weight determination. The LCMS is a technique which is done for the biopharmaceutical analysis. So nowadays we are using the Agilent, and the software includes WinLine softwares 5.2 version, WinLine software. So as HPLCMS and UPLCMS, if you see the applications of the, or the some of the salient features of LCMS, generally this is done for the molecular weight determination, and as the mass is the detector, we are estimating the molecular weight of the compounds based on the mass spectroscopy, and determining the molecular weight of the fluorescent proteins, and even for the like other analytical spectroscopic methods which is used to determine the structural determination of the genosites, that is as the glycosides. And as a pharmaceutical applications includes rapid chromatography of the benzodiazepines and identification of bile acids metabolites. Biochemical applications includes rapid protein identification using the capillary LCMS and database searching. So clinical applications includes highly sensitive for the detection of the trimipramine and thiodiazine. if you see the some of the mass spectroscopy components which are having so what we discussed earlier, which shows the again in the diagram as inlet, source, analyzer, ion detector, data system. So if we focus on the mass spectrometer, so this is the diagram. So a sequence of the diagram which explain about the proceed of the mass spectrometer: inlet, source, analyzer, ion detector, data system, and mass spectrum is recorded. So as this LCMS is the system which is the quadruple system of Q1 and Q3. So as a Q1 and Q3 quad system, general setup in the triple quad system, which includes in the next diagram is ion production, transport, filtering, fragmentation, ion filtering, ion detection. So as a part of LCMS system, still I am continuing some of the diagrams which exclusively focusing on the instrumentation or the rock diagram of the mass spectrometer and proceed of the Q1, Q3 triple quad system. That is, as we seen in the previous slides, this next slide will explain about the ionization source as four methods. And it proceed for the nebulizer collision gas, which will causes the Q1, Q2, Q3, which explain about the quadruples as a mass analyzers, and finally it will be detected in the form of the different detectors. So just now we discussed in the earlier slides. So whereas voltage parameters, next slide will explain about the voltage parameters in the quad. That is different abbreviations which we come across in the next slide. So this next slide will explain about the voltage parameters which come across in the quad MS. That includes some of the salient points which explain about the Q0, Q1, Q2, Q3. So in the form of some points, that is first one we are applying the voltage related points, which includes declustering potential DP. So it is on the orbital plate, whereas the potential which is required in the skimmer and Q0, that is the entrance potential EP, whereas CEP, that is the entrance collision potential, which we can call as collision cell entrance potential, whereas CE, which explain about the collision energy. For the collision, the amount of energy which is required is the collision energy, CE. Collision cell exit potential, CEP. Like CEP, we are having two CEP. One is entrance potential, another one is the exit potential. So these are some of the terminology which we come across in the voltage parameters are concerned with the Q0, Q1, Q2, Q3. So DP, EP, CEP. Another CEP we can even abbreviate as CXP, exit potential. So if two CEPs are there, one CEP is entrance and another CEP we can call as CXP as exit potential. And this will explain about the points. So if you see the quadruple in the next slide, one diagram of the quadruple, the basic points of the quadruple, that is, it is formed by the set of parallel round rods. So one set is electrically connected with the positive DC voltage and other one with a negative DC voltage. This is one diagram explain about the quadruple rods and additional RF voltage at fixed frequency which has an aptitude that is oscillate between the positive and the negative. One side is positive and another one is negative. So this is oscillate between the positive and negative, which is applied to the four surfaces. And related monitoring devices, which includes the singular selected ion monitoring in the next slide. So which will explain about the singular selected ion monitoring. So there are different one. One is MRM, one other one is SIM. Here it is SIM, selected ion monitoring. So that is Q0, Q1, Q2, Q3 quadruples are explained with the selected ion monitoring. Precursor ion selection in Q1, no fragmentation, passing of the precursor ion. That is, it allows the selected ion monitoring in the SIM. Whereas another mode is, one mode is SIM, another one mode is MRM. That is multiple reaction monitoring. Single reaction monitoring is SIM, whereas multiple reaction in the next slide, multiple reaction monitoring is there, that is abbreviated as MRM. So regularly we are using the regularly we are listening the word called MRM in the case of LCMS. So multiple reaction monitoring, which will in this also Q0, Q1, Q2, Q3 quadruples, which allows the all ions to pass through. Whereas Q1 selects an M plus H, that is generally in the positive mode or negative mode. Ions are there. So whereas Q2 fragments the selected ion, Q3 monitors only one daughter ion in the MRM. Quadruple ion trap. So that is Q1 selects a parent ion. Next slide will give the quadruple ion trap. So that will explain, these are some of the points which are uh, in the instrumentation point of view. Q1 selects a parent ion. Q2 fragment the selected ion. Q3 traps, then scans out all the fragment ions. Fragmentation of fragment is possible, which provide more information for the structure. Later on, identification and selection of the exit ion can be customized as per the requirement. Whereas the next one will explain about the triple quadruple, that is IQ1, IQ2, IQ3. So this will, there are different gases, curtain gas as gas flow. So curtain plate. So this will give the IQ1, IQ2, IQ3 as the exit lanes. Next slide will explain about the triple quadruple. So ion filtration based on their MZ ratio using the DC or RF voltage applied to the quadruple. So this is the diagram of the triple quadruple as a curtain gas. Next slide will explain the detector as a channel electron multiplier as SIM and photomultiplier. These are the, so in the previous slides we explained the detectors. All these slides explained previously. I'm showing in the diagrammatic point of view each one. Again, I am summarizing as a detectors. So SEM and ETP, channel electron multiplier and photomultiplier tube. So PMT, regularly we are listening the word in the photomultiplier tubes in the previous spectroscopic methods also. Here also we are getting this ETP as a photomultiplier tube and channel electron multiplier. So based on this LCMS, suppose we are having any one method, how to done the method development based on the LCMS based is just as a one or two slides I explained the method development and validation parameters as far as LCMS is concerned. So till now we discussed about the LCMS hyphenated technique, evaluation of the analytical techniques and all this. So here based on this LCMS method, how the method is developed and what are the validation parameters as far as bioanalytical method development is concerned. So you'll see in the upcoming one or two slides which explain about the method development based on the biopharmaceutical analysis. The next slide. It involves the evaluation and optimization of various stages of sample preparation, chromatographic separation, detection and quantification. Like HPLC method, so LCMS method development parameters, which includes the either of the one of the method as chromatographic GCMS or LCMS, as HPLCMS or UPLCMS, and all these the MSMS. So that is in this method development, choice of the mass parameters such as parent ion, product ion, or choice of the ionization as positive mode or negative mode. Choice of the compound parameters as the difference, that is declustering potential DP. So and collision exit potential and this collision potential, all these. Whereas choice of the gas parameters as the curtain gas, nebulizer gas, heater gas, and the collision aided destruction gas. That is the choice of the parameters, choice of the ionizations, choice of the compound parameters, choice of the gas parameters. So selection in the method development. Next, choice of the chromatographic conditions as a mobile phase, column, auto sampler conditions, flow rate, injection volume. Choice of the internal standard. choice of extraction method. So extraction methods, which includes denaturation method, solid phase extraction, liquid extraction based on the nature of the sample. Next, choice of the regression methods. So these are the based on the things. Whereas method development parameters as a flowchart, that is so mass parameter optimization, positive, negative mode. So what we discussed earlier is in the flow diagram, that is chromatographic conditions, extraction methods, that is mass parameters as voltage, gas, temperature. Chromatographic optimization as mobile phase, column, auto sampler, injection volume. Extraction methods just now SPE, LLE, and protein precipitation. Internal stand optimization. So based on the hyphenated techniques, method development in the form of the flowchart. Next, parameters of the method development in the form of the parameters as a validation parameters, precision, accuracy, and matrix effect, recovery, regression model, and stability. Different types of the stability. Generally the validation followed by the method development. Validation is the technique we already know. The validation involves the documenting through the use of the specific laboratory investigations, performance of the characteristics of the method, which is suitable, reliable, and so intended analytical application as a part of full validation. In the next slide, full validation, partial validation, and the cross validation. So next slide will explain about the validation parameters. Validation parameters. Validation is just now. So those includes different parameters as far as biopharmaceutical analysis is concerned. That is biological matrices. So selectivity and specificity, precision and accuracy. So it is intraday and interday, as we know that the validation parameters are already in the all analytical methods. So recovery and matrix effect here includes ion suppression, ion enhancement. So ruggedness and stability is already we done for the different type of the methods for the stability as benchtop stability, freeze the auto sampler, long term stability, short term stability, different types of freeze thaw stability, or the different type of the stability methods which are done as a part of the validation parameters. So in the slides, we saw the applications of the LCMS and even advantages and disadvantages are also regularly there for every analytical methods. So and hyphenated techniques are a little bit clicked methods and advanced and sensitive methods. So definitely having major advantages as a enhanced the analytical power through the combination of multiple techniques, that is as LCMS, GCMS, so and all this. So these are increased selectivity and specificity for better detection and identification of the analytes. That is, selectivity is increased and specificity is better in the detection for the identification of the analytes in the hyphenated techniques. Whereas time and cost efficiency by integrating multiple steps into a single workflow. Expanded application range across the various sample types and analytes. Depending upon the sample type and the analyte, we are expanding the application ranges and comprehensive structural information about the compounds, facilitating in-depth characterization. So of course, disadvantages are also there. So with this hyphenated techniques, that is complexity and high instrumentation costs, that is complexity and high instrumentation costs, and challenging in the method development and optimization. And so hyphenated techniques includes different type of hyphenated techniques having the advantages. So over that, the LCMS is the highly advanced technique. Generally, the major two-step process which occurs in the LCMS is, so in the liquid chromatography or HPLC, sample components are separated based on their interactions with the mobile phase and stationary phases, as mentioned in the HPLC section. Once the chromatographic separation is completed, the compounds elute from the column and are ionized at an ionization source. Subsequently, the ions compounds are introduced into the mass spectrometer for the precise mass analysis. So LCMS consists of HPLC unit as a LC with a mass spectrometer detector attached. Whereas in the case of exclusive HPLC, it allows the physical separation of the components. So MS measurements, that is mass to charge ratio of the ionic species, are so nowhere related to this. And regularly this MS, which is an analytical tool highly used in the science labs, so to determine the masses of the different compounds in the sample. Mainly this mass spectrometry, that is, we can say the word called tandem mass spectrometry. This tandem mass spectrometry along with LC as a liquid chromatography mass tandem mass spectrometry. So this will explain the identification and quantify the compounds in the mixture as well as to detect the impurities in the sample. So this mass spectrometry employing the various ionization methods, determining the masses of the molecule by measuring the mass to charge ratio of its ion. So this will create a high vacuum environment, which consideration of the specific factors. So the different ionization methods, mainly the factors which includes as far as ionization method is concerned, volume, concentration, sample phase, composition of the analyte solution. All these are the taking into the consideration of the advantages. We are using the so hyphenated techniques. So whereas the next slide will explain about the disadvantages of the technique. So that is complexity and high instrumentation cost. And another one is challenges in the method development and optimization. So that is in the method development and optimization, there are some cases. So depending upon the type of the extraction method, in some cases, extraction method selection is becomes typical. Whereas the complexity in the data and interpretation analysis and stringent sample preparation requirements and sensitivity and robustness limitations due to interfaces. So that is matrix effect we discussed in the slides, that sensitivity and robustness limitations due to the interfaces and the matrix effects. So these are the some disadvantages. Of course, disadvantages are there. So, but this is the LCMS method is a clicked method for the biological matrices estimation in the, so even proceed for the pharmacokinetic parameters for the explanation of the samples. So this will give an idea of the different various hyphenated techniques apart from the LCMS techniques. So there are different hyphenated techniques along with the LCMS, which includes is GCMS, gas chromatography and mass spectrometry, and other techniques, which includes is LCNMR, liquid chromatography, nuclear magnetic resonance spectroscopy, GCIR. There are different hyphenated techniques as LCMS, GCMS as GSC and GLC, LCNMR, and GCIR, GCNMR, and LCIR methods. So these are the different hyphenated techniques. Among that, LCMS is the so a clicked method of hyphenated technique which comes in this. And nowadays, current scenario will move on the LCMS and the GCMS. So with this, we explain about the so various levels of the hyphenated techniques as a different analytical techniques. So thank you all for the what we summarized in the different techniques of the analysis. So in the, all the slides, what we explained, it will be summarized in one point that. So in this session, we explained about the different techniques of analysis. So from the electrochemical and all these spectroscopic, chromatographic, and we move to the hyphenated techniques. And all these techniques will give the idea of the various levels of the hyphenated techniques as the LCMS. So thank you all for the opportunity.
Thank you, sir.
Sir.
So, dear delegates, do you have any questions? You can unmute yourself and you can ask.
No, it's excellent. Wonderful session.
Thank you, sir. So, any queries from the participants? So, you can unmute yourself and you can ask. Okay. Let me thank Dr. K. Ravikumar Garu on behalf of the Vasavi Institute of Pharmaceutical Sciences, so management and principal. So giving a wonderful information about the chromatographic methods on HPLC and LCMS. She has explained excellently about the mass ionization methods and the spectral information. So, and what are the different applications of mass spectroscopy hyphenated with liquid chromatography. So, with this, I thank Dr. Ravikumar Garu for having a speech on our FDP on innovative methods in analytical sciences.
Thank you, sir. Thank you, Dr. Ravikumar Garu.
Thank you, sir. Thank you for providing me
excellent
opportunity.
presentation regarding different techniques in analytical sciences.
Thank you, sir.
And very informative. And I hope that participants also had a nice informative session. And thank you once again to all the participants. And we will meet tomorrow by 11 o'clock with the other session. So thank you, sir. Thank you for your valuable time and valuable presentation.
Thank you very much.
Thank you, sir.
Thank you.
So, participants, a feedback form will be circulated in the WhatsApp group.
WhatsApp group.
In WhatsApp group in a few minutes. So you can answer that.
Right.
Okay. Thank you. Thank you, everyone.
So thank you. You can leave the meeting. Thank you.
Bhaskar sir, shall I end the meeting?
Yes, we can end the meeting.
Thank you, sir.
Thank you. Thank you.